Welcome everyone. Our speaker today is Nataša Jonoska, and she will be talking about topological problems associated with DNA nanostructures. Take it away.
Thank you very much, Teresa, and I wish to thank all of the organizers and Henry in particular for this kind invitation to give this presentation and to share some of my research interests, in particular for accommodating the schedule, because it seems that it's always difficult to find right time for everybody. So this is really nice that we can schedule this, and thank you for the invitation indeed. Before I understand that many of the talks and much of this seminar is really geared towards TDA and maybe some statistical analysis together with the TDA. So my presentation will be applications of topology, but not really geared towards TDA. So I will talk about DNA nanostructures. This is one side of my interest besides other biological questions. And I hope you find this interesting from that perspective. There will be some topology, but not different, I guess, from what you have experienced so far. So I will start with the discussion of DNA nanotechnology, and this is chemical view, I guess, of the DNA. DNA consists of two strands, right? What's important is that the strands consist of bases, nucleotides. Nucleotides have bases, and these bases come in 4 instances: adenine, guanine, thymine, and cytosine. So A, G, C, and T, as we already know. A is complementary to T, and G is complementary to C. And when they find in opposite orientation, that's very important in our discussion here, then they join together and form this double helical structure. So at the one side of the strand, there is a 5', we call it a 5' end, which has a phosphate. On the other side of the strand, we call it a 3', which has a hydroxyl group. So the strands are oriented. The hydroxyl group connects with the 5' end. So the 3' end connects with the 5' end of the opposite strand. So in some sense, you can think of this being in the opposite direction. So this directionality is important. This molecule is a molecule of life. We can see it in all living beings everywhere. And this is a segment of Rousseau's painting with all sorts of life in it. But what we will talk about will be the molecule for actually nanotechnology. So this is the first molecule that was introduced by Ned Seaman, who has been known as the father of DNA nanotechnology. This cruciform of DNA, each one of these lines consists of one strand. This is a holiday junction. As in biology, this holiday junction happens often actually during recombination processes in the cell. However, it's never stable. So this moves all around. It's never a stable molecule. However, Ned introduced asymmetry in the sequence of this molecule, such that this holiday junction now became a stable molecule, and it could be used like a Lego blocks to form other nanostructures. So I'm going to look at the mathematical perspective of this nanostructure. So what type of molecular communication is involved in molecular self-assembly when the molecules come together and build up larger structures? Are shapes of molecules result of a computation? What is the process that actually produces the given shapes? What type of structures can one make with building block components? So imagine that I have these Lego blocks sort of components. What are the structures that can arise by self-assembling these molecules? What can those structures be identified? How can be identified? How can be described? Can we design functionality or roles, and we say this Lego block will be on this part and this Lego block will be on this part, and then when they come together, they have this function and so forth during the process? And can we actually talk about the process and distinguish different processes of self-assembly? So as I mentioned, the molecule, the DNA is a double helical structure. And this is a first building block that was actually designed in a lab. So this building block self-assembles into these cruciform forms because the molecules are complementary, the nucleotides are complementary to each other. So this portion here is complementary to that portion, this portion is complementary to that portion, and so forth. So when you have design complementarity of the DNA strands in a nice way, they actually self-assemble in different structures. So this is why this self-assembly process has all these different mathematical questions that mathematicians have not really addressed. And so why DNA? And I'm going to cite here Ned, who was my collaborator for over 20 years, up to his passing, actually, about 3 months before he passed away. We actually got another grant approved. So it was really a long-standing collaboration, and I still collaborate with his lab, which is now led by Ruji Shah. So I'm gonna cite here Ned, and he actually, if he showed this slide, there will be like double of what I have reasons why DNA is a good molecule to do nanostructures. But I'm going to focus on some of them. So there's a predictable intermolecular interactions, which is the self-assembly of complementarity of the strands. So we can predict which portions of the strands can interact with other portions of other strands. There are convenient automated chemistry. Of course, the biotechnology have designed all this machinery that we can already deal with. We can synthesize, we can sequence, we can active enzymes, we can order buffers and molecules and sort of. There are enzymes, so the nature has already provided enzymes that are modifying the DNA in a variety of ways, so we can use them, we don't need to invent them. So it's a locally stiff but globally flexible molecule. So if I have a very short duplex, double-stranded molecule, the duplex of double helical structure, it's very stiff. It's really like a Lego block, like rotini, if you wish. But if it's long, then it's actually quite flexible. You can sequence and you can read the code, right? High functional density, group density. So this, you can, I didn't mention the measurements, but in one helical turn, there are about 10.4 nucleotides. So that means, well, 4 to the 10th information stored in 3.4 nanometers. So one helical turn is 3.4, 3.5 nanometers. The diameter of the DNA is 2 nanometers. So I have 10 to the 4th possibilities to store in 3.4 nanometers. And it's a prototype for many derivatives. We have used, now there are lots of different molecules that are being derived out of DNA with similar properties as DNA. What can we do with it? Well, DNA can be a scaffolding because we can design very precise nanomaterials. So it can be used for as a scaffold for other particles and proteins. And we can develop new technologies for studying biomolecules. We can use it to design targeting drug delivery because it can react in natural cell environments. And we can use it as information storage. And as I mentioned, lots of information can be packed in, even in one helical turn. And for me, it's fun. So we just do it for are just, I guess, part of our studies, and I guess all our research is really, it's supposed to be fun, right? So these are several examples of what have been done. This is the first two-dimensional array that was done in Ned's lab. There are other two-dimensional arrays that have been obtained with DNA molecules, DNA nano. So these are the other possibilities and the 3D structures that have been obtained, all of these with the DNA nanomaterial and design of the DNA. So I'm going to focus on 3D structures now and think about the molecules as three-dimensional structures. And I can think of the self-assembly as information processing in 3D. I mean, everything is happening in 3D. In information processing, we don't really have a good mathematical model for this because, well, I guess we need something like shape processing. Everything that our electronic computers do are on zeros and ones. In fact, is one-dimensional. And we're talking about three-dimensional, so 2 dimensions up, and we have no mathematical models how to deal with these things. So I'm going to talk about graph structures in 3D and how we can, how some topology questions arise in these three-dimensional structures. I will mention some algebraic structures motivating DNA origami, but maybe I'll skip that. I will probably focus at the end of my talk to DNA crystallographic structures, which is the latest development in the lab and our interests that we have. So we'll start with spatial graphs, and I'm going to show you one simple example. Imagine the vertex colorability problem, and you probably, and when I give this talk to people, to different audiences, I have to explain what vertex colorability problem is, but you probably all know. Given a graph, I want to color the vertices of the graph so that no two vertices that are adjacent have the same color. And I'm going to use 3 colors, so 3 colorability problem. And this is an example of a graph and the coloring of the graph that is 3 coloring. So in order to design, to solve this problem in a lab, and experimentally, what we do, we actually going to construct molecules, as I said, small Lego blocks, where each vertex will correspond to one of these junction molecules. So for example, this is vertex 6, right, colored with red. I'm going to have 3 instances of colors. I'm going to have this molecule with the red color here. So this is a sequence that is encoding a red color. And the single-stranded portion of the sequence that will try to find its complement in the design. So this sequence here is a red color. I can design the same molecule, exactly the same molecule, but I'm going to change the sequence to be green and another one with blue. So there will be 3 copies for each vertex. So this will be vertex 5 that has only 2. This is vertex 4 that has 4 valency, and so forth. And the complementarity will be, for example, this is complementary to this. I will have A complementary to A bar, C complementary to C bar, and B prime are the colors. They're distinct sequences, not complementary to each other, so green and red. The design is such that when I have 2 molecules of the same color, inside the sequence that encodes the color, we have actually put a sequence that is recognized by a restriction enzyme. So when the restriction enzyme, I have this full coloring, right, the same coloring of the edge, the complements are complete, right, and I have a double helical structure, and the enzyme recognizes this structure and cuts the molecule. However, when the molecule, when the 2 vertices join together with opposite colors, right? Different colors, not necessarily opposite. Then there's a bubble because these are not sequences that are complementary to each other. So they don't really form a double helical structure, but there's a small bubble in the middle. And then the enzyme cannot recognize that structure and just doesn't cleave. So the final structure.
Sorry, maybe to interrupt you here. There was a question in the chat. I think I even know the answer. Why is vertex 5 colored not green? I think you said that you should imagine that each of these pictures exists in all 3 colors, right?
Yes, every vertex will have 3 colors, right? All 3 molecules. So I will have a copy of red, a copy of blue, a copy of green, and all of them will have, each vertex will have a copy of a molecule represented in 3 colors. And I just point out the amount of vertices that the amount of molecules we're talking about. So each molecule appears in about 10 to the 15 to 10 to the 17 copies in a test tube. So there are huge amount of copies of each molecule inside the test tube. So the final structure that we get is a structure of this form, where I don't know if I answered the question.
Yes, I think you answered the question. There are lots of copies, and 1/3 of them is red, 1/3 of them is blue, 1/3 of them is green.
Exactly. So the final structure, when we form the graph, which will be 3 colorable, it will be something like this. So this is a molecule that we're expecting to get. And in here, okay, I want to point out that this was done by self-assembling. So the whole process was design these molecules, you have the graph in mind, put them in a test tube, self-assembly, put 3 enzymes to chop up the red colors that are joined together, the green and the blue, and fetch out the molecule that you want, the double molecule, the final structure. So this is a computational problem that is being solved completely by self-assembly, right? Can I color this graph with 3 colors? So there's a big question that arose from this. If natural self-assembly process that produce complex molecular shapes that we have in life, in biology, perform computation, what do they compute? Can we actually understand the problem that they're actually solving by attaining these complex structures? Because this is a very complex molecular structure that we obtained at the end. And does this computation confer a selective advantage in evolution? So the whole experiment was quite easy, to be honest, but it was difficult in the sense that we had to prove that this is indeed the structure that we got. So the way we proved it was we introduced another molecule, another enzyme that is ligation, which joined the nicks. So if I have one strand coming next to the other strand, this enzyme actually joins the nicks and forms one single strand. So we ligated the molecule, and we wanted to fetch one molecule, and this is I have colored in purple here, that traverses the graph, every edge of the graph at least once. So this purple molecule, once we fetch this purple molecule, I had the whole information in the graph in the molecule. We can sequence it, we can check that this vertex is next to this vertex, that this is the edge connecting them, and so forth. So what came out of this is, well, okay, this design is nice, and the graph has this, I can find this reporter strand, which I call it the reporter strand. Does every graph have a reporter strand? So I need reporter strands in some sense if I do want to design 3-dimensional structures and to be able to show that those 3-dimensional structures indeed exist.
Sorry, so, maybe one moment. There was another question in the chat. I think it was basically just that this person lost a bit the connection of how does the theoretical problem of graph coloring have something to do with biology. But I think like you were just explaining this for a long time, but maybe just a summarizing sentence for someone who got lost.
Well, the graph coloring has not really, there is no connection to the biology per se. What I was trying to say is that just through self-assembly and enzymatic activity, we obtained a very complex molecular structure like this that provided us with lots of questions that are essentially biological questions, the ones that I wrote at the bottom. Can we look at complex shapes of molecules, either on proteins or DNA or other structures, as a result of computation? Can we actually detect the problems that they're solving in order to attain these different complex structures that have been obtained at the end? Because they're all result of self-assembly and enzymatic activity, everything we have in life. So I'm going to go back to talk about the reporter strand. So the reporter strand can be looked at a walk in the graph that visits every edge at least once, and at most twice, of course, because we cannot traverse the edge more than twice. And the second time has to be in the opposite direction, right? If I travel to this strand, I have to traverse in the opposite direction because the DNA is oppositely oriented, and I need to be preserving the structure of the graph. So in an Eulerian graph, the Eulerian, where every vertex has an even degree, the Eulerian circuit can be an answer. So the question was, can this be done in arbitrary graph? And the problem turns out to be the variation of a Chinese postman problem, which is known to be an incomplete problem. So we're going to do mathematical design. So we're going to look at the thickened graph. So I'm going to consider the edges of the graph as flat strips, right? So the double helical structure, if I have, if I look at the double helical structure, I can just look at one strand and another strand, and this will be my strip. The rims will be oppositely oriented. So in this strip, because the DNA strands are oppositely oriented. And I'm going to look at the thickened graph, right? Which will be a graph made of these strips as a two-dimensional manifold, such that G is topologically embedded as a one complex and as a deformation retract. So this thickened graph is not necessarily unique for this G, but the boundary components, the rims that I obtain at the end, are in fact the DNA strands that I need to have, that I will eventually end up in the construction of the graph. So here's an example. Imagine this graph. So this is one f of G, and this is another f of G, different surface, different manifold representing the same graph. For this graph, however, I can only design this f of G here. I can flip this, but essentially it will be the same topological structure. So a reporter strand of the thickener is a boundary component now that traverses every edge at least once. And we can show that every multigraph has this reporter strand, actually has a boundary component that visits every edge at least once. And then Joe Ellingham and Mark Ellingham in Vanderbilt, they proved that this process of getting the optimal reporter strand turned out to be NP hard. But the fact that every graph has one was a very nice and interesting observation. And I'm going to tell you how you can prove this. It's actually not that difficult. Imagine that here's my 3-degree vertex in a graph, and I follow the purple strand. It goes this way, the green goes this way, and the blue goes this way. I'm going to introduce 1/2 turn, 5 nucleotides in my DNA, and then change the vertex. Instead of going up, my purple will go down now. And blue, instead of going left, is going to go right, and green will go up. So this is a small change in the vertex connectivity, but quite different, quite big change in the graph. So I'm going to show you how that shows up. So here's a graph, and here are two possibilities, right? So this is a planar graph, and I'm going to introduce boundary components in this planar graph following every cycle of the graph. So then this would be one possibility to construct this graph with the DNA. As I mentioned, every boundary component, you can think of it as being a DNA strand. But if I introduce this change in this vertex and in this vertex, then I end up with another, I guess, situation in the graph made of single strands. So this instead of 5, I think there are 5. Yeah, there are 5 here. I end up with a single boundary component of this surface that traverses the graph at least once. Actually, every edge is traversed twice. Everything depends on the embedding. So here I have a planar embedding 5 boundary components. Here I have, if I embed this graph on a torus, right, I am introducing one change in the vertex, and here's the embedding. I am missing 2 cycles here, one boundary component and another boundary component. This is a reporter strand. This is a reporter strand that visits every vertex at least once, and the graph is embedded in a torus. If I introduce another handle and a double torus, then I end up with the nice structure that I said, that the whole graph is traversed with a single boundary component, but the graph itself is really embedded on a torus. I'm talking about cellular embedding. Sorry, I should have mentioned that. Cellular embedding on orientable surfaces. So we can do several observations, and the final observation is that the number of boundary components depends on the, of course, on the Euler characteristic, and also on the genus of the maximum and minimum embeddable surface where the graph can be embedded cellularly. And this formula can be derived easily. Okay, I'm going to mention now DNA origami. I have about, I guess, 10 more minutes. Maybe I'll finish with some. So DNA origami was introduced in 2006. And now I'm going to switch to show you a short, maybe a video. Let's see if I can find the, yeah. So here's a video that was prepared by Sean Douglas about DNA origami. There's a long strand, this long strand, and all right, but these short strands are actually grabbing complementary to different parts of the molecule of this long viral plasmid, and joining these different parts of the molecule and form this nice structure. Okay, let me see. Go back to my slides. So this was introduced by Paul Rotemund, this origami structure. The whole idea is you have a long viral molecule, a viral plasmid, and you have about 250, 260 complementary single-stranded molecules that capture different sides of the molecule. Of course, it was the front page of Nature, smiley face. He made like 3 billion smiley face, very happy Nature issue. And this is a design, and this is the actual experimental result. It works very well. So people got crazy and started doing all sorts of nanostructures with DNA origami, as you can see, many different structures. And I can't even start. There are about, I don't know, maybe 300, 500 labs that are using DNA origami currently in their designs. So I'm going to point out something that was really relevant in topology and about this design that was done in Karolinska Institute in Sweden, and it appeared in Nature 2015, which is designed with DNA origami of meshes. I mean, if you do any computer graphics, you know this bunny. So the mathematical part was done by Malik Mohammed, who ended up in my group as a postdoc. So imagine that you have a target shape and you want to design this. The whole point is to traverse this mesh in an Eulerian circuit and design these short staple strands that will bring this little, the long strand that traverses the whole graph, the reporter strand, with complementary parts that can join different parts of this long strand in a way to form this shape. And it was very successful, as you can see, all right? So this is all the design, and at the bottom you have the experimental results, images of the experimental results. So the way that one can think about this is, how do we actually find different trails, different traversing into the graph? So if I have a vertex of degree 8, the first Eulerian path will join two edges. The first traversing will join two edges. The second one will pair two other edges, and so forth. So the pairing can be intersecting the edges, if I think about the rigid vertex, or they can be neighboring, which is often called an A-trail in topological graph theory, or they can be non-intersecting pairing, which is non-A-trail, but they're just non-intersecting. So the question that we started looking at was, what can we do about meshes or structures that are embedded on other surfaces, not on a sphere? Everything that they did originally here was on a sphere. Everything is spherical, if you can think about it, topologically a sphere, right? So we looked at the, say, embeddings on torus and other surfaces. As an example, take this K7, right? So it's K7 is intrinsically knotted. However you embed it in space, there will be a cycle that is knotted. But I can do a cellular embedding on a torus. And this cellular embedding on the torus, where I can take this purple, the purple cycle is here, then I use the red going around 3 times, and then the green goes around 3 times. But it's cellular embedding. Once I do a cellular embedding of the K7, I can actually do a checkerboard coloring. And when I have a checkerboard coloring, we have an A-trail traversing of the graph. And A-trail traversing of the graph always provides a nice Eulerian circuit. So this was very nice, and we can actually prove that all non-intersecting scaffold routings are unknotted for checkerboard colorable graph embedded in the torus. You start with an unknotted cyclic molecule, you have to end up with an unknotted structure. So traversing of the structure. So I didn't mention that, and I should have, because all of our plasmids are unknotted. So it says they're cyclic, but they're not knotted. And when we do design of the structures, we have to come up with a routing that is not going to be knotted. And we can also show that there always exists an unknotted non-intersecting scaffold routing in a closed orientable surface. And the checkerboard is really colorable graph is really the important part. So I went back and talked to Ned, and he said, well, I tried knotted. So he knotted the big long plasmid. And I produced different structures. What structures can I get if I actually start with a knotted, not an unknotted structure? So we looked at other observations, an Eulerian graph, admits embedding in the standard torus, okay? So that all its non-intersecting scaffold routing are noted if and only if it admits an embedding that is not checkerboard coloring. So if I have an embedding that is not checkerboard coloring, I can do a routing that is knotted. And for all for all n greater than 2, right, n tori, we can have checkerboard colored embeddings in n tori that have knotted non-intersecting scaffolding coloring. So this was also interesting, that even the checkerboard colorable cellular embedding can produce non-intersecting scaffold routings. Okay, and I will use my last 3 or 4 minutes just to show you. In January of last year, I organized a small meeting where we started looking at the DNA crystals, and this is my group, and we invited Luke Kaufman and Simon from the lab, and Eleni Panagiotou from and Wout Walker from Amsterdam to look at structures and crystallographic structures. So DNA crystals, this is crystals that were obtained in 2010 in Ned's lab. And I'm going to show you a couple of several crystallographic structures. Hope you don't mind me moving into okay, sharing camera. You see camera here?
Yes.
Okay, so I'm going to show you several. So first of all, I'm going to show you the standard one that you just saw an image of it. So this is an image of that standard crystallographic structure. This is the triangle that was originally formed to form that crystal. And if I look at the crystal itself, it looks like this. And as you can see, it has a very nice crystallographic structure. So the central strand is going, I have colored in red the central strand, which is a very important, I guess, meaning for us in the mathematical design. As you can see, it's a cyclic molecule in the center of the triangle. So this was original. Then the lab came up with this structure, which is the tiny little structure, and you can't even see this, but there's a central molecule here, which consists of 3 nucleotides from one junction to another, very, very short. And if I look at the crystallographic structure, this is the crystal that is being formed from that. Okay. The crossings of the sides of the triangle are opposite than the crossings of the sides of the triangle in the 7 nucleotides. So the original was 7 nucleotides from junction to junction. This is 3 nucleotides from junction to junction. The central strand is very difficult to see. So, okay. So that's, and here's a 5 nucleotides between junction and junction. There's no triangle. Actually, the 5 doesn't form a triangle. It just doesn't like to be a triangle. And it does form a structure, a crystallographic structure, but it doesn't form a triangle. And if let me see if I can find a nice location to see it. As you can see, oh, maybe here, there's this row, and also there's this opposite going from through the screen. It's more or less 2-dimensional. All of the DNA is going 2-dimensional. There's nothing that goes in the 3rd dimension here. However, if I look at the original 7 nucleotides, as you can see, there are actually 3 dimensions, right? Going in all 3 dimensions, and the structure, the crystallographic structure, has DNA in all 3 sides. So one goes this way, one goes this way, and one goes through the screen. Okay, so the lab came back and said, well, there's these opposite crossings. So here are the opposite crossings of the triangle, one here and one here. I don't know if you can let me just pull this off. As you can see, this is one. And let me see, show you another one that is left-handed, maybe this one. So as you can see, this is on top of that. And here I have this below that one. So the crossings are opposite in the triangle. So the lab came back and said, why is this happening? And can we explain this? So we went back and did our modeling. And I'm going to go back to my slides and show you. Okay, I'm going to skip this part. I can address that. But this was work with the lab. Simon was a postdoc in the lab, and Van is a postdoc in my group. And then we also worked with Masahiko. The paper appeared at PNAS in April this year. So we looked at the number of nucleotides between junctions when triangles form, when triangles don't form, when are left-handed, when are right-handed. And we looked at the central strand, and the whole design was concentrated on the central strand. The DNA was really we considered the DNA duplex, the double helical structure, as a cylinder, right? And these cylinders are really, this is how they form the triangle in 3D. So it's a standard model where we made sure that the diameter is 2 nanometers, that the helical turn is 3.4 nanometers. I will not go through the details. I just want to show you that you start with the standard helical curve, and then you rotate the helical turn in a way such that the curve starts and ends on the same axis, right? And the cylinders start from here, move to there. Imagine that these are the boundary of a cylinder here of the double helical structure. We move it to here, then we rotate it, and it ends up in this kind of structure. It turns out that when you rotate this curve around the axis, you end up with rotations of the curves around the axis in a variety of ways. And we can show that the smoothness of this angle here is the important part. It has to be either maximum or minimum. Of course, you can introduce the complementary strand at that whole. But if this is one, for example, curve for the central strand. If I rotate, I will get another central position of the central strand. So we measured the central strand, we looked at the smoothness, we looked at the triangle cavity between the cylinders, and also the cylinder tilt, because the cylinder has to offset out of the plane in order to get the three-dimensional structure. And so here are the curves that we obtained for different rotations. And we observed that we have to have a tilt that is greater than 2 nanometers, so the diameter is completely off the plane. We have to have either local maximum or local minimum for the smoothness or the sharpness of the curve at the junction between the two curves. And we looked at the maximum cavity in the triangle. Of course, there's also another consideration, which is the complementary strand. The complementary strands cannot collide, and they had to be larger than experimentally measured value of 0.62 nanometers. So we ended up with this. So this curve, if I am in this location, so the green is the smoothness, right? The blue is the tilt level of the cylinder, and the green is the distance, the cavity inside the cylinder, inside the triangle. If I have a positive cavity for n equal 3, for 3 nucleotides from junction to junction, then I get this nice cylinder look position. They're left-handed. If I have n equals 7, and I'm in this position here of the curves, then I have this nice position of the cylinders. Rotating the curve anywhere around, you get some mesh of this or mesh of that in the n equal 3 case. So we went down and we do the computation, and we measured with experimentally obtained tensegrity triangle, and we got this result, and it matched very nicely with the experimental result. I just wanted to point out about the left-handed and the right-handed triangle here. The cylinders in, for example, in 17 nucleotides going from here to here, from junction to junction, we have right-handed crossing. And if I have 14 nucleotides from here to here, I have a left-handed crossing of the cylinders and left-handed triangle. So the chirality turned out to be really dependent on the number of nucleotides from one to the other. And there's nothing we can do, but we can explain it mathematically, because those are the curves that had to be satisfied. So these are the experimental results on the left, and the mathematical comparison on the right. And this last column here are the superimposition of our design of the curve, the central strand, with the measurements, experimental measurements of the atoms of the structure that was obtained by the lab. For n equal 15, it's similar to n equal 5. There is a crystal, very two-dimensional crystal in some sense, but you cannot get the triangle, and we can show mathematically that it's just impossible to get the triangle. I just want to point out that all these structures are raising zillions of mathematical questions. What are the unit cells on all these crystallographic structures? What type of invariants can distinguish building blocks that build large 3D constructs, crystallographic or not? Can we characterize and the strand topologies that can appear within these structures? And the recent paper by Mohammed from the group that I joined last year, Mohammed El-Hamdadi, Wout Molwalker, and Masahiko Saito was just posted actually last week, introduced the notoid quantal invariants for these notoid structures that we are looking at. This is just to finish. DNA nanotechnology has been growing. This is a book that appeared last year. I'm not advertising myself here. I'm just pointing out that this book is, we actually negotiated with Springer to be open source. So it's open source. You can look at it online free of charge. So lots of mathematical questions there, zillions. And thank you very much. And I'm sorry it took me a bit longer than I expected.
Thank you very much, and that's no problem. It was super interesting. Let's please all unmute ourselves so that we can clap together.
Okay.
Nice. So let's start with questions. Who has a question? Maybe while people are thinking what questions they have, I can start with one. Yeah, it sounded super fascinating to me that it's you can basically construct materials like knitting or so, just that it doesn't take time to knit it all. You can just let the biology do its work. And I was thinking, can you, for example, also then make crystalline materials that have certain properties that you want? Like, for example, an auxetic material where if you push from two sides, it doesn't extend in the other dimension, but it shrinks in the other dimension. That
Well, those are all of the questions that we are actually looking at in the lab. How can we design crystallographic structures? So this one example here that I skipped over, yes, you can design. Actually, this was, let me explain how you can do that. Let me see, find one nice example here. Okay, so imagine that I have a crystal structure like this, and I'm gonna introduce a little hairpin on the sides of this triangle, right?
Mhm.
You design it in a specific manner. So this hairpin can then, by introducing a new strand, open up and expand the structure, or take the strand out and close it up. The hairpin will shrink the structure. That theoretically can be done. All of those designs are in the work of we have used these crystals to introduce new particles in variety of ways. We have tried to design a Janus crystal, which is a combination between, actually the lab has done that, where you mix two different units in construction of a crystal, one and the other. So this, which is very difficult experimentally to do in general, to design a crystal with the different units. And but I would like to point out that the idea about this was actually to do a computation and to the aperiodic crystal, design of an aperiodic crystal. And to design an aperiodic crystal, you can imagine that I can, in each one of these sides, I can encode a function. I can encode a computation, a function. I can design strands that correspond to 0, 1, or 2. So I can look at the ternary computation, ternary function. So for this would be a buildup of a function x, y, z, x plus y plus z plus 2 mod 3. So I can have green, which is 0, red 1, blue 2, for example, 3 distinct triangles, and build up the structure. For this function, you get an aperiodic structure of this form. So this is algorithmic design of aperiodic structure in 3D. And this is something that is a first step towards designing an information storage material at a nano level, when you algorithmically can access each one of these units, or position the units in the right manner. Of course,
Yeah.
we haven't done this yet. This was just the theoretical design.
That's fascinating. Okay, then there's some people thanking you for the talk. And also a question, could you please repeat what you meant by the smoothness of an angle, what angle you were considering, and why it is important for that angle to be smooth?
Oh, let me see. So this is the design of the. So the central strand, which is the strand that this strand here, which I pointed out with red in my camera image that I showed you earlier. So this central strand is traversing the cylinders, right? But it's going from one cylinder, crossing to the other, and then crossing to the third, right? So this central strand, it turns out that, let me see if I have, here's the central strand, right? The connection between this move from one cylinder to the other cylinder has to be smooth in here, or the way that the way that the n=3 is designed, it has to be as sharp as possible, because it comes from one cylinder and immediately crosses to the other. It doesn't go around, but it crosses immediately to the other strand. So it has to be either the maximally sharp to cross from one cylinder to the other, or it has to be smooth to go around one cylinder and then crosses to the other. So those are the two extremes. And it turns out that the two extremes, when it's maximal sharpness, we end up with left-handed triangle. And when it's as smooth as possible, we end up with a right-handed triangle. So the chirality is uniquely determined by this, the shape of these curves of the central strand molecule that we designed, well, the model that we designed.
Nice.
I don't know if I explained that.
Yeah, the person who asked the question already wrote thanks.
Oh, okay.
And there's another question which I think should be easy to answer. Are the knots you mentioned in the last slides the same knots as from knot theory?
Yeah, I'm talking about knotoids, right? So here we're going to talk about, because none of this, all of these curves that I mentioned here, where am I? So these are all chopped, right? Here I have a nick. This is a one, well, it's written strand. Well, here maybe it's a more accurate. So they're all curves that are part of the structure, but they're not closed curves. So yes, knotoids that I mentioned and linkoids are exactly the knotoids and linkoids from knot theory.
Okay, perfect. Then Dave, please ask your question.
Yes, when these molecules, like crystal-like molecules, are assembled in the lab, from what you're saying, you're taking these building blocks consisting of junctions of 3 molecules, and they're of a certain, they have certain length and all to get the correct structure. When you're assembling them in the lab, do they, from what you said earlier, you have a test tube and they're put in the test tube, and maybe that's not a simplified version. Did they always assemble the way you want, or some fraction of them assemble the correct way? Or do they come out flawed in some way? Some are nice, some aren't so nice.
Oh, of course, some are nice and some are not so nice. It turns out that these crystals, actually, it takes a long time to assemble. There are only 2 nucleotides, so the junction, the connectivity here is only 2 nucleotides. So there are only 2 nucleotides that are complementary to each other. The assembly takes a long period, so the molecules are all put together and slowly cool down, and cool down to maybe 4 degrees Celsius. And it takes about a day, I think, 24 hours to 36 hours. It's slow cooling for them to assemble. And they're actually pretty good in assembling these structures. The crystals that we obtained, or that the lab obtained, let me see if I can get back to some of these images. Here, these crystals are actually 200 micrometers. 200. So as you can see, these are large crystals by chemical observation. They can be seen, some of them, by naked eye.
Oh.
So it turns out that the molecules, of course, everything requires lots of salt and lots of positive charge, because DNA is negatively charged, but they want to do this. So the whole process is really just letting them cool down and self-assemble in a very slow manner.
Yeah. Good. Thank you.
Jacob?
Hi, I was wondering if there's like an obvious reason why linkoids would be preferable to periodic links.
Well, because the strands are really linkoid, right? Let me see. So this image, these are all linkoids. Multiple strands that are not connected to each other. They're not closed curves.
But in like the bigger structure, it looks like there was like some repeating patterns.
Yes, in crystallographic structure.
Okay. And so I was thinking maybe periodic links would be an option to model those like repeating patterns.
Yes, periodic links would be, but having a nick, it actually introduces sometimes small variations that are not necessarily obtained if I connect them.
Okay. Okay.
And we don't have a nice way of design of really characterizing those. But yes, periodic links, Eleni has actually started looking at this as periodic links, and she has introduced the Jones polynomial invariant for periodic links.
Okay.
But we need to have an invariant that will characterize these structures particularly. And it turns out it's not easy to compute none of those that we had.
You're saying like the linkoid, nothoid invariants are harder to compute?
Because, well, structures are a little bit elaborate. Exactly.
Oh, yeah, I see. Okay, thank you.
Right. We had already so many great questions, so I think I can end the recording here. And I just want to thank you again for this particularly nice talk.
Okay, thank you.